All right. So B6, B7, B9, and B12, these are going to correlate very heavily with many of the vitamin cofactor and coenzyme roles when we look at protein metabolism. So this lecture really focus on what is the B vitamin involved in and what is it's a cofactor for. Because when we, after this exam, we're going to pick right back up with protein metabolism, and you're going to see how all of these carry forward. So these two introductory lectures of the B vitamins will help you definitely in protein metabolism. So be sure you recognize the B vitamin and the name of it. So B6 is your pyridoxal, and your active form is your PLP. Your B7 is your biotin. B9 is folic acid if it's synthesized, and that is the oxidized form. And then folate is the form of B9 that is found naturally in the dietary sources. And B12 is cobalamin. So let's jump into B6. So the name of B6, pyridoxal, active form, pyridoxal phosphate, known as PLP. And one of the primary things that B6 is known for is that it's involved in greater than 100 different reactions, all in amino acid and protein metabolism. So when you think of protein metabolism, greater than 100 reactions, I want you to be thinking about PLP. What is the primary physiological role of B6? And that is going to be your PLP to support protein and amino acid metabolism, which is also why you're going to see a lot of B6 come back up once we get into protein metabolism. So the other thing that you can recognize, however, I want you to know the PLP, that there are other forms that the body will toss it around back and forth. However, the most prominent and most important one, especially with the metabolism of our amino acids, is the PLP. However, in nature and in our physiological body, we do have different forms of it where it changes hands. So that's what that is demonstrating for you. So I want you to pay attention right here in yellow. So what are the known causes that deplete B6? And these are important because if you look at this simple list of, it's 3 bullet points, but there's 3 more points. These are the people that you have to be worried about for being B6 deficient. So the categories are anybody that's on an exogenous corticosteroid. That is somebody that is anybody autoimmune is almost always on regular rounds throughout the year of corticosteroids. I have a good family member and she has lupus, and I want to say 2 to 3 times a year she'll have an episode, and her doctor, her rheumatologist, they just put her on a systemic shot, and they just pump her body full of exogenous corticosteroids, and then they'll do her on a dose pack, and they'll do an oral one for like another 5 to 6 days. And it unfortunately, it's a very nasty disease. Then it takes a lot of anti-inflammatories, but I worry about her B6. So exogenous, meaning it comes from outside the body, and corticosteroids, that's your anti-inflammatory steroid. Birth control is notorious for depleting B6. And then because B6 is a water soluble vitamin, we need to be thinking about loss in diuretics and alcohol. Diuretics are going to increase the amount of loss to urine. So any B vitamins are going to have a higher risk of becoming urine. And then alcohol, we talked about, that is inhibition of your ADH, and you do not conserve water. If you do not conserve water through the aquaporins in your filtrate and nephron, then you're going to lose a lot of your minerals and your B vitamins. And then the hypertension medication, definitely. How many people are on blood pressure meds? There's a lot. The population is very rampant for blood pressure meds. And that, again, that alters the kidney's ability to regulate the resorption. So I want you to be thinking of exogenous corticosteroids, birth control, blood pressure medications with your diuretics, and alcohol. Okay, so what two catabolic pathways require B6? What about transaminase and when does this occur? This is a board's question. They almost always are going to ask you about transaminase, and then we dive into that pathway in more detail after next week when we get back into protein metabolism for the next set of material. So right here, the two catabolic pathways of the B6 are going to be transaminase and tryptophan degradation. Another word for degradation is catabolism. So that's the breakdown of amino acids and or the amino acid tryptophan. So what is transaminase? I want you to know this first sentence. It is the first initial step of amino acid catabolism, and that removes the NH2. Transaminase is the first step that removes the amino group from an amino acid. We learn in more details what that pathway is, but for now, B6 is the vitamin dependent cofactor for your transaminase, the first step of catabolism that removes your NH2 group. Okay, so when does this occur? That occurs when we want to break down an amino acid, hence the first catabolic breakdown step. All right, so how is B6 correlated to oxidative stress, to glutathione, and what is the pathway? We're going to review this in more detail once we get into the second segment, so I'm just going to give you this clue. We're going to learn the pathway because the pathway is the transsulfuration pathway. And guess what? Our very important sulfur amino acid cysteine, which is the precursor for our intrinsic potent antioxidant tripeptide glutathione, are dependent on two steps that require B6, and that is your transsulfuration pathway. So if you are low in B6, then you have less efficient synthesis of glutathione. If you don't have enough glutathione, you are at potential susceptibility for oxidative stress, because glutathione by itself is a reducing agent that protects against free radicals by donating a hydrogen. And secondarily, two glutathiones become built into our glutathione peroxidase that protects us against hydrogen peroxide oxidative stress. So B6 is vital in the ability to generate the cysteine and the glutathione, and that is your transsulfuration pathway. All right, so how does B6 correlate to heme? And what is the step? So B6 is going to be the cofactor that takes succinyl-CoA plus glycine. Remember back to week 3 when we learned iron. The first step to synthesize heme is the precursors succinyl-CoA plus glycine. That catabolic enzymatic, or not catabolic, excuse me, that enzymatic reaction that combines succinyl-CoA with glycine in the first step to synthesize our heme requires B6. So if we're low in B6 and we cannot do that first step, then guess what? We are more susceptible to not having enough heme. Therefore, you have to think, how would it clinically correlate? How would it clinically present if my patient has low heme? Then we'd have to remember, what are our heme cofactors? Hemoglobin, carrying oxygen, catalase, that protects us against our hydrogen peroxide, so that's increased oxidative stress from another mechanism. It's also required for that thyroid peroxidase. Remember, iodine plus tyrosine through a heme-dependent thyroid peroxidase makes thyroid hormone, and then we need it for our electron transport chain and our cytochrome C. So B6 is now part of your brain when you have to think about heme and you've got succinyl-CoA plus glycine plus B6. All right, so what intrinsic antioxidant could be infected? I just said that that's your catalase. So catalase is heme dependent, which is now B6 and heme dependent. And your outcome is anytime you have a low availability of your antioxidants, that is susceptibility to oxidative stress. Susceptibility to oxidative stress. You have to be worried about their DNA, their blood fatty acids on their P2 and their folding of their proteins. How is B6 correlated to mood, anxiety, and joy? And what about sleep? So look here, what did we say about B6 PLP? It's involved in more than 100 reactions of the amino acid metabolism. And amino acids are precursors. They build proteins, and enzymes are proteins, and neurotransmitters need amino acids. Hormones need amino acids as precursors. So check this. We need B6 in the synthesis of neurotransmitters, both dopamine, that's your feel good, that's your joy and happiness, pleasure neurotransmitter. And then how about serotonin? You think mood and digestion are going to be correlated with serotonin? Absolutely. And then serotonin converts to melatonin at night when your circadian rhythms are changing. So you need B6 to convert serotonin into melatonin. So if someone doesn't have sufficient amounts of B6, don't you think they might be susceptible to some mood and depression and maybe some insomnia? Absolutely, because that's how our body supports the synthesis of these important hormones and neurotransmitters. We're going to learn lysine. The carnitine shuttle is very important, and we'll learn that in detail in the protein metabolism. I'm letting you off the hook. I'm not testing you on the carnitine shuttle on this exam. However, don't forget it because it's coming forward and you will need to know it for the protein metabolism. So you can make a little check, protein metabolism. We're going to look at the nucleic acid thymidine when we go farther down. So just know that PLP is also important in this step, and we learn that in more detail going down. All right, so what about the tryptophan? So we said that the B6 is part of your tryptophan. If you remember, tryptophan is one of your aromatic ringed amino acids, and I'll show you the pathway. So let's take a look at this because this is really cool. So you are not drawing this out. You need to know the things that I teach you that correlate to what I want you to know from a global perspective about these pathways. So when we look at the tryptophan in B6, I want you to notice that we are looking at it in two steps. So when we convert serotonin to melatonin, this is tryptophan de novo synthesis of melatonin. However, when we have tryptophan and we are breaking it down instead of using it as a building block, right? An amino acid can do two things. An amino acid can stay its amino acid and then it goes into a protein or it goes into a hormone and it stays the tryptophan or it stays the amino acid while it goes into the precursor role to building blocks. The alternative of an amino acid is going to be it's broken down. What did we say about the first step? B6 dependent transaminase is the first step of an amino acid catabolic step. So you would need it right here, not demonstrated in this pathway. However, it is. So once tryptophan continues to go through several breakdown steps, catabolic steps, we get to this right here. And this is what you need to do. You need to know kynurenine. Okay, so when your body has sufficient amounts of B6 as PLP, it will properly synthesize as the breakdown product and continues. We will generate an important molecule called kynurenine. Kynurenate is a marker that B6 is plentiful. Now, the alternative, the problem. So what happens if there's not enough B6? We can test it because what the body will generate when there is not enough B6 to generate kynurenine as it continues in its breakdown, we get this molecule right here, xanthinurate. So B6 deficiency will show up as this molecule, xanthinurate. And when we have plenty of B6, when we break down tryptophan, we will get kynurenine, and that tells us we have an efficient amount of B6. Now, if we take one more step further, if you look a little further, so this is a different picture of how the pathway, so this is two different authors presenting the pathways differently. So your tryptophan is going to be going through hydroxytryptophan. That's where we get to serotonin. This is your synthesis pathway. And then when we want to break it down, it goes through kynurenine, which is generated if we have enough B6, or we get this aberrant xanthine when we have deficient. However, the reason that we want to have kynurenine is because we can actually generate the important B vitamin derivative, nicotinic acid. So tryptophan is able to synthesize de novo synthesis. If we have enough B6, we can generate the very important reducing agent B vitamin derivative B3. Okay, as nicotinic acid. That could be a board's question. Tryptophan can synthesize the B vitamin nicotinic acid. I'm testing you to know it's B6. They would not. Okay, so what is the name of B7? Now, I showed you before I go to B7 that these are the next few slides. When you go through this, you do not have to memorize the pathways. I'm showing you here association. B6 transaminase, okay? B6 tryptophan catabolism, melatonin synthesis. You need to know your xanthinurate for deficiency and your kynurenine for okay amounts. That's simply a different picture. So B6, here is the pathway in your transsulfuration. There are 2 steps. You're not responsible for that. You just need to know that B6 is transsulfuration and the generation of cysteine and glutathione. I'm showing you the pathways so that you can see it. You are not responsible for drawing them out. Again, this is the pathway for our heme. So we take a succinyl-CoA plus the glycine. It requires B6. So that is what you need to know. And then here is your, we'll look at this for thymidine. This is your 510. So B6 is responsible to help us make the nucleic acid thymidine. We learned that going forward. And then here is another picture for your tryptophan and showing us that tryptophan is the amino acid that can give us the nicotinamide NAD reducing agent, de novo synthesis, if we have sufficient levels of B6. And then here is the synthesis of dopamine requiring B6. And then here is your carnitine. I told you you're not responsible for yet at this week. Okay, biotin. What is the name of B7? Well, guess I just said it, it's biotin. What are the 2 physiological roles that B7 is going to participate in? So the 2 things you have to recognize are biotinylation. Guess what that ation means? That means that they're taking a biotin and they're putting it on something. That's basically a conjugation. So biotin will be put onto, look at this, next key concept, that is going to be part of your histones. This is an epigenetic, genomic responsibility, physiological role of your B7 biotin. So we have biotin-dependent enzymes that do biotinylation at the level of the genome. It opens up the genes and it inserts or it takes off a biotin. Biotinylation, epigenetic gene modulation. The second role is going to be this big funky word, carboxylase. So if you see here, we have four carboxylases. You do need to boards associate biotin is a carboxylase. Okay, biotin is a carboxylase. Now, what do you see? 1, 2, 3, 4. Guess what that means? Do you see here? If you recall, we have a dehydrogenase complex, B1, B2, B3, B5, ALA. That's your board's question. Dehydrogenase complex, B1, B2, B3, B5, ALA complex, okay? However, there was 3 isoenzymes. The enzyme itself was virtually similar, except it was different in that it had a different substrate. This is the same thing, a biotin, which has a functional cofactor role to assist the carboxylase enzyme. A carboxylase is a B7, biotin dependent enzyme. So we have, look here, lipid, glucose, macronutrient. Lipid, glucose, macronutrient. What category is that? Those are macronutrients, aren't they? B7 as biotin dependent carboxylase is going to be vital in the metabolism of carb, fat, and proteins. All macronutrient metabolism depends on B7 as biotin carboxylase. All right, so I skipped up here. Biotin, interestingly, and this is just a quick clinical pearl, I'm not testing you on this. The dietary bioavailability of biotin is fairly ubiquitous. It's found in a lot of different things. However, when we have a healthy gut, we also have the benefit that some of our bacteria will also do us a favor and a symbiotic relationship, and they'll make us some biotin. So we do have to, this might be a board question, so doc this one. Avidin in raw eggs can inhibit dietary biotin. And it does it right here with this funky word. The biotinase is inhibited by the avidin. So, and that comes from raw eggs. So associate biotin, avidin, dietary biotin, B7, with raw eggs. This used to be a problem when there was like a big fad with workout, the what are they called? Oh my gosh, competition people, weightlifters, there we go, weightlifting. They used to drink raw eggs. I don't think that's a thing anymore, but there was this whole population of competitive weightlifters that were drinking raw eggs, and then they found out that it was this avidin because they were all biotin deficient, showing up with some biotin deficient symptoms. Okay, so the dietary source that can inhibit it is raw eggs. The enzyme that this food inhibits is biotinase, and the molecule is the avidin. Avidin inhibits the enzyme. How does B7 carboxylase correlate to macronutrient metabolism? So I already showed it to you, so let me show you a pretty picture. So here's a wonderful article. If you are interested in biotin, you can read this full text. This is a phenomenal article. But look here, when we have the catabolic steps, each one of these is going to be their metabolism, not catabolism, metabolism. Some is a synthesis, some is a breakdown. However, just know that macronutrient metabolism depends on biotin across the board. So here you can see fatty acid synthesis. That'll be your acetyl-CoA carboxylase. We look at that in protein metabolism. Your branch chain, leucine, and your branch goes through a special biotin. We'll learn that in another few slides. Your other two branch chains, isoleucine, valine, plus our vitally important methionine and threonine, those go through a reaction. However, you should already recognize that, and I'll show you that in a minute. And then glucose as well because of the carboxylase. But this is actually a gluconeogenesis, and we learned that for exam 3. Here's a pretty picture. This is another really great article. And inside the cell, you can see where the genomic biotinylation occurs, as well as in the mitochondria where our carboxylase is acting. Okay, so when we talk about B7, we need to review what succinyl-CoA was. So if you don't fully remember succinyl-CoA, then we are going to do a quick review, because succinyl-CoA is going to be generated from, this is a review, you need to know this for your exam. Ready? Isoleucine, valine, methionine, threonine. Isoleucine, valine are your two branch chain amino acids that are part of this pathway, plus methionine, plus threonine. All of those amino acids get catabolized, which is another word for they are broken down. When they are fully catabolized, we get succinyl-CoA. Why was succinyl-CoA important? Number one, heme plus succinyl-CoA, heme is generated by succinyl-CoA plus B6 plus glycine. So we need it for heme. The second reason we need succinyl-CoA is because it's going to be part of the intermediate molecules of your TCA cycle. And then part of the TCA will turn the succinyl-CoA into complex 2 of your electron transport chain in order for your body to synthesize ATP. So we need succinyl-CoA as a precursor for heme. We're going to metabolize it in the TCA cycle, and we're going to generate succinate dehydrogenase from the electron transport chain. So that's energy. So succinyl-CoA is very important, and we get the succinyl-CoA from the catabolic breakdown. I have another picture somewhere down here. Okay, so isoleucine and valine break down all the way, and there is a biotin-dependent reaction. However, if you look at methionine, methionine breaks down and it uses the same enzyme, the biotin carboxylase. And here's our threonine. That's your biotin-dependent carboxylase. So when we are looking at the generation of the succinyl-CoA from isoleucine, valine, methionine, threonine, we have to recall that we need biotin carboxylase to generate the succinyl-CoA. Okay, so let's see. What other 4 B vitamins? Okay, so let's do this all the way. Why is succinyl important for energy, heme, and where does it come from? So we just answered that. How does B7 correlate to the generation of succinyl-CoA? Because isoleucine, valine, methionine, and threonine all share that biotin-dependent carboxylase. The biotin-dependent carboxylase. What are the 4 amino acids that generate it? Isoleucine, valine, methionine, threonine. All right. What are the 4 B vitamins that are required? Ooh, now that's a layer, isn't it? Guess what? Branched chain keto acid from last week required the B1, B2, B3, B5 ALA dehydrogenase. What's it called? Branched chain keto acid dehydrogenase. So all 3 branched chain amino acids have to go through that step in order to get to the further down steps. So when you look at the catabolic breakdowns, up here is your branched chain keto acid dehydrogenase, B1, B2, B3, B5, and alpha lipoic acid. A whole bunch more steps happen, a bunch of conversions, and then we get to the biotin dependent step. And this is the one that is shared, right? It's shared with threonine, methionine, and then both isoleucine and valine. So I'm testing you in the way to pull together the information that the branched chain keto acid is your B1, B2, B3, B5 alpha lipoic acid. Now, further down in the breakdown, we're adding in biotin as part of the steps before we get to the succinyl-CoA. All right, let's take a look at B12. So I skipped B9 and we'll come back to it and you'll see why in a minute. There is a method to my madness. Okay. Where is my B12? We're probably going to run over just a few, but it should be close though. Okay. B12. So first thing you got to know is B12 is cobalamin. There are 2 active forms. The 2 active forms in the body are going to be adenosyl, and methyl. Adenosyl and methyl. Okay. Adenosyl and methyl. Now, you can dietarily take other forms if it's in a supplement. If you see hydroxycobalamin is very popular. However, hydroxy is not one of the active forms. You will. Cyano is another one. Cyano gets a lot of bad rep. So I don't know, the literature is mixed. The point is, your body will take the dietary and or the supplemented B12 source, and it will put it into the active form that it needs. If it needs more adenosyl, it will convert the dietary supplemental form into adenosyl. If it's methyl, and as long as you don't have an inherited problem with that, which we learn in a few, then you will use and convert it to the methyl. Okay, so B12, unfortunately, is one of the B vitamins that is kind of specific for diets, and there are very specific people who are at a high risk of deficiencies. So the first thing you have to recognize is that your B12 is not found in plants. That means you have to eat seafood or meat or chicken in order to get the B12. So who do you think would be the first risk group? Your vegetarians and your vegans, or somebody who doesn't eat a lot of meat. So we have to be thinking about that when we're worried about our B12 risk groups. The other thing that if you recall from pathology and physiology, is that in order to absorb your cobalamin, your B12, you have to stomach release your intrinsic factor. Your intrinsic factor binds to the cobalamin, and then it has to make it all the way through the entire football length of the intestines to the ileum, because board's question, the B12 is absorbed in the ileum. The B12 is absorbed in the ileum. So that means the intrinsic factor, which binds to B12 for absorption, that's probably a board's question. You also need to make sure that B12 with intrinsic factor is going to bind and go all the way to the ileum. So what about all of our people who have gastric bypass? They don't have an ileum because it was surgically removed. So gastric bypass is one of our big risk factors for having a B12 deficiency because the ileum is removed. Our vegetarians and vegans are a big risk of deficiency because you don't get natural formed dietary cobalamin in plant-based dietary sources. Here's the next really big one, and this is important. This will come forward again. If you have low stomach acid, which is hypochlorhydria, and a lot of people who eat antacids or are on GERD medications, so that's an important one. Hypochlorhydria is also induced by an H. pylori infection. These people do not efficiently break out the cobalamin from the food matrix, and therefore it's not available to be able to bind to intrinsic factor, because hydrochloric acid breaks down meat. Meat contains B12. If you have hypochlorhydria and you are not sufficiently generating stomach acid, then the B12 will not be broken out of the food matrix from your animal sources, and that is a problem, and that will be a B12 deficiency. Then, of course, any disease that is not, if you can't absorb it, such as celiac or IBD, then you could also have a risk. So be sure you are familiar with those. All right, so this is why I jumped B9. Guess what? I'm dragging you back to succinyl-CoA because there's more to the story. Succinyl-CoA has got to be like one of my favorite molecules because it's so important. It's right up there with heme. Okay, so let's go back to our two branch chains, isoleucine, valine. And we know that methionine and threonine are also part of the mix. So when we are looking at this, so we already saw this, and now that very last step, this is the key word, I want you to know this, the very last step to generate the vitally important succinyl-CoA is your adenosyl B12 step. That's where your body uses the physiological active form of cobalamin as an adenosyl. So let's take this complex thought. Okay, complex thought. What does transaminase do? What did we learn up here about transaminase? Transaminase is a B6 dependent first initial step of amino acid catabolism. Transaminase breaks down the first step of the amino acid to allow it to get further catabolized to generate the end product, which we use for metabolism. So the first step in branch chain breakdown is B6 transaminase. Okay, write down the B vitamins, B6. Okay, branch chain amino acids. First step, B6 transaminase. What happens next? The next step of the branch chain is branch chain keto acid dehydrogenase complex. So the second step of your branch chain, B1, B2, B3, B5. Okay, so collectively, does that mean step 1, B6, step 2, B1, B2, B3, B5? So sequentially, B1, B2, B3, B5, B6. Okay, cool. Are you following me? Few more steps, cool. The body's doing its thing. And then we get to our B7 carboxylase. Okay, so is that B1, B2, B3, B5, B6, now B7? Wow, that's a lot of B vitamins. Oh, wait, we're not done. That's right. Let's keep going, because before we get to succinyl-CoA from our branch chains, we have to use adenylyl B12. So succinyl-CoA from isoleucine and valine, which generates your succinyl-CoA, leucine does not. That's why when I say the 4, isoleucine, valine, methionine, threonine. Okay, don't screw up thinking it's the leucine, because that's it's a different pathway. So isoleucine valine branch chain breakdown gives us succinyl-CoA. B1, B2, B3, B5, B7, and B12. So how many B vitamins does it take to get succinyl-CoA? All of them except folate. All of them except folate. There's not a specific B9 reaction. So if somebody is fatigued, right, and you're thinking maybe they don't have enough B vitamins, you think it might be affecting their succinyl-CoA and their heme? Yeah, absolutely. What do you give somebody with fatigue? You pump them full of B vitamins. All right, so let's see. How many B vitamins? I hope you got all of them. Be sure to name them. Now, lastly, before we keep going, follow me on this blue box. Blue box, the B7 dependent carboxylase. If you notice, I'm not making you learn the name of it. Know that it's a carboxylase, and I'm happy with you. B7 dependent carboxylase has 4 isoenzymes. They act in different ways. Right now, you need to know that it's a carboxylase, and this one is shared by isoleucine, valine, along with threonine and methionine, because this is its pathway. Methionine goes through its jazz, it gets down to the biotin dependent carboxylase, and then it keeps going, and it gets down to the step where it needs adenylyl B12, and then boom, you've got succinyl-CoA. Here's your threonine, it does its thing, it goes through its catabolic steps, and then it gets to the shared biotin carboxylase step, boom, keeps going, and then we get to the adenylyl B12 step, boom, now you've got succinyl-CoA. Okay, so that's how you need to learn those. Now we have to jump back up and look at folate. Folate is important, and folate is a unique B vitamin. So the first thing that you need to know is that folate is going to be foliage. I want you to associate the word foliage. The reason for that is very little B9 and folate is going to be found in dietary meat. It's going to be mostly in plant-based, especially green leafies, hence the root word foliage. When we're looking at so that means somebody who eats all meat and potatoes isn't going to have sufficient B9. So somebody who's a vegetarian and doesn't eat any meat or seafood is going to be deficient in B12. Somebody who doesn't eat meat is going to be, or excuse me, so if they don't eat foliage and they only eat meat, they're going to be likely deficient in folate. All right, so what is the active form of B9? B9 is tetrahydrofolate. Tetra, 4 hydrogens on a folate. Tetrahydrofolate, 4 hydrogens. What's it called when you put hydrogens that you can remove? Guess what? This is a redox vitamin, very potently involved in redox reactions. So tetrahydrofolate, THF, is what you need to know, and that's because it has the redox potential, which makes this a unique molecule. No other molecule besides right here, biopterin, these two uniquely have the redox potential of 4 hydrogens. They lose them 2 at a time. So we learn that in more detail when we get into the folate, or excuse me, into the protein metabolism. And right now, you need to recognize that the tetrahydrofolate is unique for 4 hydrogens. It's the active form of folate, and that unique property is shared with this, biopterin. Now, this is the next things that you absolutely need to know for this upcoming exam, and this is a board's question. Folate pool is called a 1-carbon transfer. Folate metabolism, you have to sustain the folate pool. I'll explain that in a minute. And folate metabolism is associated physiologically with 1-carbon transfers. 1-carbon transfers is B9. So the other term for this is called a methyl. A methyl is a CH4. It's a CH4. You'll see that going forward. So folate, 1-carbon transfer. The 1-carbon transfer is also a methyl transfer. All right, so who's at risk for low B9? We said that somebody who doesn't eat roughage, greens, and plant-based sources. The active form, tetrahydrofolate, that's 4 hydrogens, and that is called fully reduced tetrahydrofolate. The other molecule is biopterin. So how does this correlate to the folate pool? So now let's take a look. When you look at this slide, you can see here that there are different forms. You see folic, you see folinic, and you see methyl tetrahydrofolate. And then if we come down a little bit further, I think I have another picture. Okay, here's another one. All of these are different forms. So we've got dimethylglycine, 5,10-methylene. Okay, you've got folic, dihydrofolic, tetrahydrofolate. You do not have to memorize all of that. You need to understand that the folate pool is the bioavailability of all of these. Each one in its, we call this a folate derivative, keyword, folate derivative is what we are supplying in all of the different active forms of tetrahydrofolate, and each one has a unique biochemical pathway that it supplies as a cofactor or coenzyme. So if you don't have one that gets replaced back into the pool, the folate pool, you will end up with negative downstream effects, and it will back up all of the bioavailability. So what is the folate pool? It's making sure that all of the bioavailable forms of tetrahydrofolate are available and being recycled and regenerated, going through all of the different reactions that each one of them is responsible for. So that is your folate pool. And it's physiologically important because if you get backed up on one of them, it will have negative effects. So you always have to be refilling and regenerating the folate pool. The methyl transfer, okay, so let's look at a methyl transfer. The methyl transfer, remember, a methyl is going to be a 1-carbon transfer. It's what it's known, that's what folate does as tetrahydrofolate. So your methyl group, I'll show you that when I have the picture for going forward. So I'll show you that in a second. Okay, let me get into the methionine cycle. All right, we have to learn, we're going to do the methionine cycle in a, okay, so here's your, here's the slide for your methyl group as your CH4. Definitely star and check this slide because we're going to learn all of this when we look at the homocysteine homeostasis. So we still got a little bit more to go, but hanging in with, we got about 10-15 minutes. Okay, so when we are looking at SAMe, you have to know what SAMe is. SAMe is S-adenylmethionine. S-adenylmethionine. And that is important because SAMe is going to be the universal methyl donor. And this is a B9 and B12 dependent reaction, as we see down here. So if you cannot generate SAMe as the universal methyl donor, you can end up with significant mood and anxiety disorders and symptoms. So know the word methyl transfers, that 1-carbon transfer. What is SAMe? S-adenylmethionine. And then how does this correlate to B9? It correlates to B9 because that is one of the vital functional roles that it's going to involve. And the The cycle is the methionine cycle. It's called the methionine cycle. Methionine cycle, and we learn that in detail in the next segment. So B9 is directly correlated with mood because of the methionine cycle. B9 derivative is directly correlated with cardiovascular health, CVD, cardiovascular disease, cardiovascular health, because of the B9 dependent generation of homocysteine. So we're going to learn that the methionine cycle is going to support heart health because of homocysteine, and it's going to support mood and anxiety, depression, because the same pathway also generates that SAMe, S-adenylmethionine. So be sure to refer back. Here's what we're going to learn in the next segment, so you can refer back to the picture. Here's another picture just showing it in a different way. What B9 derivative is associated with DNA synthesis and can impair RBC formation? That might just be an exam question. That sounds like a really good exam question. And we're going to learn that in detail going down here. But if you refer back, our 5,10-methylene tetrahydrofolate is the B9 derivative directly correlated to anemia. Directly correlated to anemia. 5,10 methylene tetrahydrofolate through the UDP is the B9 derivative that is directly correlated to anemia and impaired red blood cell formation. All right, so let's dive into the methionine cycle because it can be a little heavy. Another one. Okay, so pay attention to the cofactor, which is one of your B vitamins, the name of the pathway, and what is being generated. Remember, if we're going to be healing a patient, we need to be concerned about a person that has cardiovascular health because of our hyperhomocysteine, which we're going to learn in a minute. And we know that hyperhomocysteine is directly caused by a B9 and B12 deficiency. And we know that if you're B6 dependent and you have oxidative stress, you are going to be a worse damage. So we need to learn these so that we know what we're doing when we're supplementing and how to support it. Okay, what two pathways intersect for homocysteine homeostasis? I hope you see the two big bolded words. Homocysteine homeostasis is dependent on the intersection of methionine cycle and transsulfuration. Methionine cycle, transsulfuration. What is the health risk associated with hyperhomocysteine? If you have a poor methionine cycle because of deficient B9 and or B12, you are going to have hyperhomocysteine. A normal individual has normal basal levels of homocysteine because they have a sufficient B9 and B12 methionine cycle. If you have hyperhomocysteine correlated to B9 and B12 deficiency, you are at risk for clot, thrombus, atherosclerosis, and cardiovascular disease. So what B vitamins would be directly correlated to cardiovascular disease, clot, and cardiovascular, clot and thrombus? B9 and B12. What about the mental health factor? What did we say that the same pathway generates? It generates our SAMe. At the same time that we recycle homocysteine and prevent hyperhomocysteine, that exact pathway also gives us, keyword, universal methyl donor, which we need as SAMe, S-adenylmethionine. And this is highly correlated in the mental health, mood, anxiety, and depression. Also been very studied in some of the schizophrenia and psychosis as well. So why? Why is the health factor of homocysteine and mental health as SAMe correlated to B9 and B12 is because of the shared pathway. It's where they intersect, the methionine cycle and transsulfuration. Okay, so what are these pathways and which one is correlated with each? So next super important concept, I hope you actually might already recognize what I'm asking for. Your transsulfuration. Transsulfuration is going to be your B6 dependent pathway. That's going to give us our glutathione and our cysteine. Our methionine cycle is our B9 and B12, is B9 and B12. So if we are going to properly, so homocysteine is sitting here right in the middle. That means this is how you look at it. Homocysteine, when the body needs SAMe, it's going to send the homocysteine being recycled this way with our B9 and B12 dependent pathway. Homocysteine will go this way into the SAMe. Or when the body needs oxidative stress and or cysteine, the opposite will happen. It will direct the chemistry of homocysteine into a different pathway. Depending on the tissue's needs will depend on what pathway it's going down. That's why we call it an intersection, because homocysteine has to be shared between these two pathways. However, in order for homocysteine to properly be metabolized in whatever direction our body needs it, we need B6 when we go down transsulfuration, and we need B9 and B12 when we go up into the methionine cycle for SAMe and homocysteine being recycled. So which one is which? We just talked about that. How does the body generate SAMe? What is SAMe referred to, and what B vitamins? I hope that this is you've heard it a couple times. How does the body generate SAMe? It recycles homocysteine in the methionine cycle. What is SAMe referred to? That's your universal methyl donor. Universal methyl donor. Okay, universal methyl donor. And then what B vitamins are needed? That is your B9 and B12 dependent pathway. How does the body generate glutathione? What are the precursors? How does this correlate to B6? And how about to transsulfuration? Again, it's how the body works. Transsulfuration uses B6. Got it? It generates cysteine, and then cysteine, glycine, glutamate. Cysteine, glycine, and glutamate make glutathione. Those are your precursors for glutathione. So what you've learned extra today, you already knew how important glutathione was and what the precursors were. Now you know it requires B6 and it goes through the transsulfuration pathway because of homocysteine being converted. Very good. All right, clinical pearl. How does supplementing with NAC increase your oxidative protection? Guess what? If you give the body NAC, N-acetylcysteine is going to go right down the transsulfuration pathway and be a precursor to make that glutathione. So when you take NAC, guess what? Your body puts it down that B6 dependent pathway into transsulfuration to synthesize glutathione. All right, how does the methionine cycle correlate to B9 and methyl transfers? Now let's get into the methionine cycle in more detail. Here's a really cool picture. You are not required to draw this out. You have to understand the concepts that I've demonstrated. So here's the short version of it. Here's the side one, that's your transsulfuration. So what we are going to look at is the methyl transfer. It's complicated, so I did my best to show you the pictures. So here is the picture. CH3 methyl group. So we are here. How does the methionine cycle participate in methyl transfers and one carbon transfers, CH4? So methyl B9 is the active B9 that starts it. So methyl, meaning the methyl group, CH3, is on B9. So here's what happens. B9 hands off the methyl group, has a methyl transfer, and it gives it to B12. It gives it to B12. So methyl B9 transfers the methyl group, and it forms methyl B12. So now this is no longer methyl B9. Now this is just tetrahydrofolate. That step just added tetrahydrofolate back into the folate pool. Now tetrahydrofolate can go act in another pathway. While this transfer happen, your B12 scooped up the methyl group. So now our B12 is now called methyl B12, methylcobalamin. This is the next really important step. What happens is the methylcobalamin is going to put the methyl group on homocysteine. That's another methyl transfer. The methyl group was from B9, got put on B12. Now B12 with the methyl is going to give it to homocysteine. Guess what just happened? We just recycled homocysteine out of the blood. What do we not want? We don't want too much homocysteine because it causes clots, thrombus, and atherosclerosis. So what did B12 just do? It just added a methyl group onto homocysteine and it turned it into methionine. What just happened? Methyl transfer happened and was put onto homocysteine. Homocysteine plus the methyl group is now called the molecule, the amino acid, methionine. So when we start with the methyl transfer, it started with your methyl B9, your methyl folate. It then transferred it to your B12. Your B12 transferred it to homocysteine, and you end up with the recycling of homocysteine, and that is now methionine. So if you take a look at that and now come back here, okay, this explains it. Follow the methyl transfers, and then homocysteine is methionine. And if you look at it again this way, what are we doing? We're transferring the methyl group. The methyl group from B9 goes onto B12, it goes onto homocysteine, and we are regenerating the methionine. So homocysteine, once the homocysteine gets the methyl group, is methionine. And so methionine is the last one with the methyl group. Guess what? What is SAMe? S-adenosylmethionine is the methionine with the methyl group. This is one more step. It converts to your SAMe. So the methyl group being put onto the methionine not only recycles homocysteine to prevent our cardiovascular disease, it also generates the SAMe so that we can have our universal methyl donor. All right, why folate metabolism? Why is folate metabolism as tetrahydrofolate and methyl B9 important for health? Because you need it to prevent mood, anxiety, and disorder through SAMe, and you need it to recycle homocysteine. You have to prevent hyperhomocysteine, and that's done through folate metabolism's one carbon transfer. What happens to homocysteine when the body performs the B9 methyl transfer? It becomes methionine. It becomes methionine, and one step further is the SAMe. What other B vitamins need to be bioavailable for the B9 tetrahydrofolate methyl transfer? That's your B12. You cannot successfully recycle homocysteine and form SAMe if you don't have both B9 and B12. If you don't have the B12, which is part of the enzyme methionine synthase, there's nothing for B9 to give it to. So both B9 and both B12 as methyl B9 and methyl B12 are required in order to prevent cardiovascular disease and prevent hyperhomocysteine. What is MTHFR? I will save that, I think we can review that in the, let's see, 5,10-methylene. I think you guys are probably on overload. We can do all of these in the protein metabolism. So I want you to stop right there. We'll stop at the heart healthy. Okay, all of these here are showing you how the transfer goes. So I want you to learn that really good, because as you can see, the story continues. This comes up when we look at so many different diseases and we analyze metabolic syndrome, and when we analyze cardiovascular disease, atherosclerosis. All of this is important in order for you to understand some of the bigger diseases and syndromes as we move into. So let's see. The questions about MTHFR, I will delay, okay? The only thing I want you to recognize about 5,10-methylene, because I do have a quiz question, So just know that we'll learn the details once we get into protein metabolism, because it comes back up. So 5,10 methylene tetrahydrofolate forms UDP to make thymidine. Because that's going to be important, because that can be a cause of anemia. So 5,10 methylene tetrahydrofolate for your quiz is going to be important for the formation of the UDP, and the end result is thymidine. The remaining questions on MTHFR and methyl trap and 5,10 methylene, outside of that simple key concept that I just told you, thymidine, will be delayed for our protein metabolism. And then here, all I did down here was show you a couple of my favorites that I use all the time. This is a doc that does intramuscular shots, and you can walk in, it's $10. It goes right into the glute muscle. You walk out in 10 minutes, $10, 10 minutes. Definitely, he has been instrumental in helping some of my health as I refilled my B vitamins and my stress, and he's fabulous down in Central Avenue. Okay, that was a really long lecture. I know I'm a little bit over our time for today. And let me stop.